Hi, it's Miss Nicholas. First, let me apologize for the sound of this scratchy voice. I've got a little bit of that sinusy crud going on. So if you'll bear with me for just a little bit, I want to talk to you about neurotransmitters and drug actions. First thing we need to do is to review just a little bit to make sure that we're all on the same page with this discussion. You should know the structure of a neuron by now, and you should be familiar with the fact that the axon is that long part of the neuron that comes down and ends at the axon terminal or the synaptic knob. Same thing, just different names for it. And we have these vesicles that are stored down in the synaptic knob or in the axon terminal. And these vesicles that we're going to be talking about are going to contain chemicals known as neurotransmitters. So the space between, of course, is the synapse. You should remember that. Okay, so the synapse is the space. So that makes this the presynaptic neuron, and this one is the postsynaptic neuron. So the presynaptic neuron releases neurotransmitters into the synapse where they bind to receptors on the postsynaptic cell and then cause an action to occur. All right, so now let's talk a little bit more about what neurotransmitters are. Neurotransmitters, in order to be considered a neurotransmitter, must be a chemical that is produced within the neuron. Once it's produced in that neuron, of course, it's going to be found in the neuron and released from that neuron at the axon terminal when that cell is stimulated or depolarized. You should have talked about by now about that cell becoming depolarized and the calcium channels opening at the axon terminal. And when those calcium channels open, it causes those synaptic vesicles to release the neurotransmitter. All right, so once that neurotransmitter is released, it binds a postsynaptic cell. That cell must have receptors for that neurotransmitter, and then it causes something to happen in that cell. Once it has been released and it's bound to that receptor, an action is going to occur in that postsynaptic cell, and then that neurotransmitter must be inactivated, and we'll talk in a few minutes about how that occurs. If you take that same chemical, this last bullet here, if you take that same chemical, that neurotransmitter, and apply it to a neuron, it has to have the same effect as if it were released to from a presynaptic neuron. Don't worry too much about that. That's just saying that regardless how that chemical is released, it should have the same effect on that postsynaptic cell. All right, so that tells you what a neuron is. Now, let's talk a little bit about what it does. A neurotransmitter may be excitatory. Now, in order for it to be excitatory, it's going to be released by the presynaptic cell, bind to receptors on the postsynaptic cell, and when it does so, it causes sodium channels to open, and those sodium channels then, of course, are going to cause sodium to rush into that postsynaptic cell, and the cell membrane becomes depolarized. If it becomes depolarized enough, then an action potential is triggered, and that cell causes an action to occur. Now, other neurotransmitters may be inhibitory. That should tell you that it's going to prevent something from happening. So typically when a neurotransmitter binds to a postsynaptic cell and it is inhibitory, it either opens calcium channels or potassium gates. Chloride channels or potassium gates. If it opens those chloride channels, of course, chloride is going to rush from the extracellular environment into that cell. Now, chloride is a negatively charged ion, so it's an anion. You should have heard that already. All right, when those anions, those chloride ions come into the cell, they're bringing those negative charges in, and the cell is becoming more and more negative. Or what might also happen is that the potassium channels open, and the positively charged cations, the potassium ions, are going to leave the cell. So those positive charges are leaving the cell, but either way it's causing that cell to become more and more negative, whether you're taking potassium out or you're sending chloride ions in. You're causing that cell to become more and more negative. So it's becoming hyperpolarized, because remember, in order for a cell to become depolarized, what has to happen? The cell has to become less negative. All right, so when these two actions are occurring, or either one of these two actions are occurring, it's going to inhibit that postsynaptic cell from doing its job. So some neurotransmitters are inhibitory, and that's the way they work. Now, there are other neurotransmitters that do both. How can one neurotransmitter have different actions? Well, it depends on what the postsynaptic cell is and what type of receptors are found there. For example, you should have heard of acetylcholine already. Remember, acetylcholine binds to receptors on the postsynaptic cell in skeletal muscle and causes those muscle cells to contract. So that is excitatory. It excites or causes those postsynaptic cells to become excited, and they do something. But when that acetylcholine binds to, when that acetylcholine binds to cardiac muscle cells, then it is inhibitory, and it slows down the depolarization and therefore slows down the heart rate. Alrighty, moving on. Some examples of neurotransmitter actions. Acetylcholine you've heard of already. Of course, acetylcholine is going to do what? It's going to cause voluntary muscle cells to contract. Norepinephrine and epinephrine, you should be familiar with these. If not, just the common name for these two together is adrenaline. And you know what adrenaline does to you, right? It gives you that rush. Okay, so it causes a wakeful or arousal feeling. Dopamine, once again, voluntary muscle and emotional arousal. Serotonin. Serotonin has several different functions. You can see it's related to memory, wakefulness, sleep and temperature regulation, emotion. Serotonin is sometimes called the happy neurotransmitter because it promotes an overall feeling of well-being. And then GABA, gamma aminobutyric acid. Now you know why we call it GABA, right? It inhibits motor neurons. So if it inhibits motor neurons, it's going to be antagonistic to acetylcholine. All right, there are many more other neurotransmitters, but these are some of the more common ones and some that you may hear about. Now, if we're going to have those the neurotransmitters released into the synapse, they eventually need to be taken away so that they're not causing a constant action or a constant inhibition on that postsynaptic cell. So in order to inactivate those neurotransmitters, that typically is going to occur in one of 4 different ways, okay? So the first is simple diffusion. So once the neurotransmitters are released into the synapse and they bind on that postsynaptic cell, then they simply diffuse away, okay? So they diffuse out of the synaptic cleft where they can't act on that receptor anymore. So that's pretty simple. The second way is that you have enzymatic degradation, or you have to deactivate or break down that neurotransmitter. One that you will hear about if you haven't already is acetylcholine esterase. Now remember, the ASE on the end of this should alert you to the fact that this is an enzyme, and its substrate, or what it acts in or acts on, is the first part of that word, which is acetylcholine. So acetylcholine esterase breaks down that acetylcholine after it has bound to those receptors and caused that cell to depolarize. Acetylcholine esterase breaks it down to acetate and chlorine. All right, so the third way that we can have inactivation of neurotransmitters is that the astrocytes, which are glial cells, are going to bind to those bind to those neurotransmitters and break them down and remove them from that synapse. And the fourth way is reuptake. Now reuptake, to take back into. So these neurotransmitters are going to be taken back up into the presynaptic cell by an action known as endocytosis, to take into the cell. Once that is done, then of course it can be recycled and used again. It's no longer in the synapse, so it's no longer binding to those cells or those receptors on the postsynaptic cell. Okay, so now that we know a little bit about neurotransmitters, let's talk about how some of the drugs that you may commonly hear of are going to act and how they're tied into these neurotransmitters. Now, remember we talked about these drugs needing to be broken down or taken back up into the synapse. Well, if we can manipulate that, then we can look at using these actions to change depressive modes. All right, so this has to do with a branch of medicine known as neuropharmacology. And it's a branch that deals with the effects of drugs on the nervous system, and especially drugs that mimic, enhance, inhibit, or act a lot like the neurotransmitters. So a few of these drugs, and you may have heard of, are selective serotonin reuptake inhibitors. Now think about what I said, selective serotonin reuptake inhibitors. Remember, reuptake, to take back into the cell so it's no longer binding to the receptor. If you can prevent those neurotransmitters from being taken back up into the cell, then that serotonin is going to stay in the synapse. And serotonin, being that happy chemical, that mood elevator, is going to stay in the synapse, stay bound to those postsynaptic cells, therefore elevate mood. And that's the action of some of these common drugs, Lexapro, Prozac, Paxil, and Zoloft. The second class of drugs we'll talk about are the monoamine oxidase inhibitors. Monoamine oxidase inhibitors. Okay, monoamine, that should tell you that these are proteins, okay? Oxidase, ACE is an enzyme. So some of these neurotransmitters are monoamines, okay? That means that they're made of a protein, and the monoamine oxidase breaks them down. So if you break down a neurotransmitter, remember, then it no longer can do its action. So these drugs, monoamine oxidase inhibitors, prevent these neurotransmitters from being broken down. So these are going to prevent the breakdown of epinephrine and norepinephrine and dopamine. So these are Marplan and Nardil and others, but they prevent those neurotransmitters from being broken down because they inactivate the enzyme that breaks them down. Once again, if you're not breaking them down, the neurotransmitters are staying in the synapse. The third class we'll talk about are tricyclic antidepressants such as Elavil and tofranil. I'm sorry, I told you I can't talk today. These are going to impede reuptake. So they work very similar to the SSRIs, and they keep those monoamine neurotransmitters in the synapse. And the benzodiazepines, some of these you've probably heard about as well. Librium, Valium, Xanax, Ativan, Klonopin, and Restoril. These are going to inhibit the release of excitatory neurotransmitters. So if you're not releasing excitatory neurotransmitters such as epi, norepi, serotonin, acetylcholine, dopamine, if you inhibit the release of those, then these drugs have a sedative or hypnotic calming effect. All right, so all of these drugs have what in common? They affect neurotransmitter action. All right, so the last thing I want to talk to you about or tell you about is this link, which takes you to a website. And at that website, you're going to look at the effects of different drugs, different narcotics, alcohol, marijuana, amphetamines, cocaine, uppers, downers, you name it. So you're going to actually take these little mice and drag them into a chair, and from that chair they go over to this little machine that's going to analyze what those drugs are doing to their brain and how they're being affected. So take a few minutes and go over that. If nothing else, you will find it very informative, and you'll learn something about the way that drugs affect your brain and how they affect neurotransmitters. Have a good day. Talk to you soon.